Alright everyone, welcome back to the channel. Before we dive into today's fascinating and slightly icky adventure, I genuinely want to send out my best wishes to each and every one of you watching. Seriously, I hope you're feeling fantastic today, wherever you are in the world, and that you and your loved ones are staying happy and healthy. Taking care of ourselves is so important. Now, get ready, because today we're going to become microbial explorers. We're going to peek into the invisible world that's literally all around us, and even on us, by learning how to grow bacteria from everyday surfaces. Yes, those tiny little creatures you can't see with your naked eye, we're going to make them visible. It sounds like something from a sci-fi lab, but I promise you, you can do this safely at home with some surprisingly simple stuff. It's a fantastic way to understand just how much life is sharing our space. Think about your day. You touch your phone, open doors, grab the remote control, pet your dog, maybe handle some money. Every single time you touch something, you're potentially picking up and leaving behind millions of tiny hitchhikers, bacteria. Most of these bacteria are completely harmless, some are even helpful, and a very few could cause problems if they get into the wrong place. But normally, they're just part of our environment, living their microscopic lives. The cool thing is, we can actually collect these tiny neighbors and convince them to multiply into big enough groups that we can see them. They form little spots or patches called colonies. To do this, we need to give them exactly what they love, a warm, cozy place with all the food they could ever want. That's where our special growing medium comes in, often called nutrient agar. It's like a superfood jelly for bacteria. You can buy pre-made agar plates online or sometimes find kits in toy or science stores. If you're feeling adventurous, you can even make a very simple version yourself using beef broth powder, gelatin, though agar powder is better if you can find it, and water, boiled and poured carefully into clean containers with lids, like small, clear plastic containers with lids that snap on securely. Safety is super important here, which we'll talk about constantly, but the basic idea is we're making a bacteria buffet inside a sealed container. Once you have your agar plates ready, and they should look like clear jelly in the bottom of your containers, it's time to go on a microbe safari. This is the fun part. You need something to collect samples with. Cotton swabs, like Q-tips, are perfect. But you can't just use them dry. You need to give the bacteria a little moisture to help them stick to the swab. So, dip your cotton swab into some sterile water. Boiled and cooled water is good for this, or bottled water. Just get the tip damp, not soaking wet. Now, think about the surfaces you want to investigate. What looks clean but might be teeming with life? What seems obviously dirty? Your imagination is the limit. Gently but firmly rub the damp cotton swab over the surface you want to test. Roll it around. Make sure it picks up whatever invisible microbes are hanging out there. Maybe swab your computer keyboard, especially between the keys. How about your phone screen? That thing goes everywhere with us. Swab a doorknob, the kitchen sponge, that one might be scary, a light switch, the bottom of your shoe, your own unwashed hand, and then your hand after washing it thoroughly. Maybe even swab a piece of fruit skin or a coin. Get creative. Remember to label each swab clearly or remember which one came from where, because things are going to get interesting later. Now comes the crucial transfer step. Take your first swabbed cotton swab, loaded with invisible microbes from, say, your phone screen. Gently, without pressing too hard, wipe it back and forth in a zigzag pattern across the surface of the jelly in one of your prepared containers. You're basically planting the bacteria seeds onto their food source. Try not to dig into the agar, just glide over the top. If you press too hard, you might break the surface. Once you've inoculated that plate, immediately put the lid back on securely. Do not open it again. This is vital. We want to grow what we collected, not let new stuff in from the air or let anything out. Repeat this process for each different surface you swabbed, using a new plate for each one. Use a fresh, damp cotton swab for each new surface to avoid mixing your samples. Label each plate clearly on the side or the bottom with what surface you swabbed, e.g., phone, doorknob, washed hand. Masking tape works well for this. Once all your plates are inoculated and sealed, you need to put them in a warm, dark place to encourage the bacteria to grow and multiply. A cupboard or a closet usually works perfectly. Room temperature is generally okay, but a slightly warmer spot, like near a water heater, but not dangerously hot, can speed things up. Now, the hard part, waiting. Patience is key. It usually takes at least 24 to 48 hours, and sometimes up to a week, to see significant growth. Resist the urge to peek constantly. Every time you open the lid, you risk contaminating the sample or releasing microbes into your environment. After a couple of days, carefully take a look at your plates without opening them. Hold them up to the light. What do you see? If bacteria were present on your swab and they liked the food you provided, you should start seeing spots appearing. These spots are called colonies. Each colony usually starts from just one or a few bacterial cells that landed on the agar and then multiplied into millions or billions of their clones, forming a visible clump. This is the magic happening. It's like invisible cities springing up on your jelly landscape. Don't expect them all to look the same. That's one of the coolest things. You'll likely see a variety of colors, shapes, sizes, and textures. Some colonies might be tiny white dots. Others might be larger, creamy yellow blobs. You might see fuzzy, spread out growth that looks like mold, which are actually fungi, not bacteria, but they can grow too. Some might be bright pink, orange, red, or even shiny and metallic looking. Some might be smooth and round, others might be wrinkly or have jagged edges. This incredible diversity is because different types of bacteria have different appearances and preferences. The colony from your kitchen sponge might look wildly different from the colony from your washed hand. The plate from the bottom of your shoe might be a whole jungle. This visual difference is a simple way scientists start to identify different types of microbes. Think about what you're seeing. That big, fuzzy colony on your remote control plate? That might be a fungus like mold. Those small, round, white colonies. Probably common skin bacteria like Staphylococcus. Those shiny yellow colonies? Could be a type called micrococcus. The ones that look slimy and spread out? Might be Pseudomonas, often found in damp places. The plate where you swabbed your hand after washing it hopefully has fewer and smaller colonies than the unwashed hand plate, showing that soap and water really do remove a lot of germs. The phone screen plate might be surprisingly busy. Phones are notorious germ magnets. The coin might have very little growth, as metal surfaces can sometimes be less hospitable. Observing these differences teaches you so much about where bacteria like to live and how effective our cleaning habits really are. It's a direct window into the microscopic ecosystem we interact with constantly. Remember, seeing growth doesn't automatically mean something dangerous is there. Most environmental bacteria are harmless bystanders. But it certainly makes you think twice about biting your nails or rubbing your eyes without washing your hands. Safety cannot be stressed enough throughout this entire experiment. We are growing microorganisms, and while most are harmless, we don't know for sure what we've collected, and some could potentially cause illness if they get into our bodies. That's why we treat these growing plates like precious, but potentially hazardous treasures. Never open the plates once you have sealed them after inoculating. The lid stays on permanently. When you observe them, hold them up, look through the clear plastic or glass. Never sniff them, obviously. When you are completely finished observing your microbial gardens and you've taken all the pictures or notes you want, you need to dispose of them safely. Do not just throw them in the regular trash. The best way is to fix them, essentially kill everything inside. You can do this by pouring a generous amount of household bleach solution, about 1 part bleach to 10 parts water, over the agar inside the closed container. Wear gloves and eye protection for this. Carefully lift one edge of the lid just enough to pour the bleach in. Do this outside or somewhere easy to clean and minimize splashing. Let it sit for several hours, preferably overnight, soaking in the bleach. The bleach will kill the bacteria and fungi. After this treatment, you can safely tape the lid shut securely and dispose of the entire container in your regular household trash. Wipe down the surface where you did the bleaching with more bleach solution. Wash your hands thoroughly. This ensures no live cultures escape into the environment. So, why do this experiment? What's the point besides making cool or gross looking blobs? It's about awareness and understanding. Growing bacteria from everyday surfaces makes the invisible world visible. It concretely shows you that microbes are everywhere, all the time. It demonstrates the power of multiplication, how a single cell becomes a visible colony. It highlights the importance of basic hygiene. Seeing the difference between washed and unwashed hands is powerful. It sparks curiosity about the diversity of life forms we share our planet with, even the super tiny ones. It's a foundational microbiology technique made accessible, showing you how scientists discover and study bacteria. It makes abstract concepts like germs and contamination tangible. And honestly, it's just incredibly cool to see life growing from something you swabbed off your own doorknob. It connects you directly to the hidden biological activity constantly happening around you. You become a citizen scientist, exploring the microworld in your own home. Imagine you're one of those bacteria landing on the agar. You've been scraped off your comfy home on a phone screen or a doorknob, stuck to a weird cotton thing, and then smeared onto this vast, strange, jiggly landscape. But wow, it smells amazing. It's like landing in a field made entirely of your favorite food. So you start eating. And eating makes you grow. And when you get big enough, you split right down the middle, becoming 2 bacteria. Those 2 eat, grow, and split, becoming 4. Then 8, 16, 32, and on. Before long, there are billions of you, all clones, piled up together in a giant mound, the colony that we can see with our eyes. All that growth started from just you and a few buddies who happened to land in that spot. That's exponential growth in action, and it's why a tiny sample becomes visible so quickly under the right conditions. It's also why infections can take hold fast if harmful bacteria get inside us and find a welcoming environment. This experiment is just the beginning. Once you understand the basics, you can design your own investigations. How effective are different hand-washing techniques? Wash for 5 seconds with water only, 20 seconds with soap, or use hand sanitizer, then swab your palm. Compare the plates. Test different cleaning products. Wipe half your kitchen counter with one cleaner and half with another, let them dry, then swab identical areas on each half to see which cleaner left fewer bacteria behind. Does the inside of the fridge really have fewer bacteria than the countertop? Swab and see. How clean is your toothbrush holder? What about the water from your tap versus bottled water? The possibilities are endless. Just remember the golden rules. Label everything clearly. Use sterile technique as best you can. Boil water, clean containers, fresh swabs, never open the growing plates, and dispose of them safely with bleach. Always wash your hands thoroughly before and after handling anything related to the experiment. Respect the microbes. They're fascinating, but treat them with caution. Seeing those colonies appear is genuinely exciting. It's like revealing a secret map of microbial life in your own environment. Some plates might be almost empty, which is interesting too. Why did nothing grow there? Was the surface truly clean? Was it too dry? Did the microbes there not like the food we offered? Others might be covered in a riot of different colored colonies, showing a huge diversity. Each colony type potentially represents a different species of bacteria or fungus, all living together. It's a miniature ecosystem on a plate. Take pictures, draw what you see, note the differences. Compare the growth day by day. You'll see colonies get bigger, change color slightly, sometimes even merge together. It's a dynamic, living display. This hands-on experience teaches you more about the microbial world than any textbook description ever could. It transforms the abstract idea of bacteria into something real, visible, and intriguing. So, there you have it. We've journeyed from the invisible world on your phone to visible colonies thriving on jelly. We've covered how to set up your agar plate safely, how to collect samples like a microbial detective, the importance of patience and careful observation, the stunning diversity of what grows, and the absolutely critical safety steps for disposal. This experiment is a powerful reminder of the teeming life we coexist with, usually without even knowing it. It highlights the importance of simple hygiene, sparks scientific curiosity, and gives you a real sense of how microbiology works. It's amazing what you can discover with some simple materials and a sense of adventure. I hope you give this a try. It's truly eye-opening. Just remember, be safe, Be curious and never underestimate the power of the tiny. Thanks so much for watching and exploring the microscopic world with me today. Wishing you all a fantastic day filled with wonder and, of course, lots of clean hands. Stay healthy, stay curious, and I'll see you in the next video. Take care.